All right, and welcome to our lecture on Chapter 16 on the innate immunity. So immunity is the ability to fight off pathogens by the body and to prevent disease. There's two main parts of immunity. There's the innate side and the adaptive side. The innate side is a generic attack, and it is always there. So even before the pathogen enters into the body, it's there and it's ready to provide protection against a disease. Whereas adaptive immunity is more specific towards a specific type of pathogen. And that takes some time to build. That will be our focus in our next lecture. Our lecture today will be going over innate immunity. And there's two main defenses. We have the first line of defense and the second line of defense. The first line of defense is preventing the microbe from entering the body in the first place. So the skin, mucous membranes, and the normal microbiota of the body all serve to help stop or prevent the entry of microbes into the body. And the second line of defense is inside of the body, and it is there, always ready to provide an attack against any microbes that enter into the body. And these include some of the immune cells, natural killer cells, phagocytic cells, and they're already there ready. Inflammation, fever, and antimicrobial substances are all generic attacks against all types of microbes. So the first line of defense of the innate immunity, again, is preventing microbes entering into the body. And that includes the skin, mucous membranes, and resident microbes. The skin, as you recall, the outside layer of the skin is the stratum corneum. The stratum corneum is the very thick layer of dead cells and keratin. And so this provides a physical barrier to pathogens. And it is also very dry. And so this also adds to the barrier aspect of the stratum corneum, as a lot of bacteria cannot survive in the dry environment of the stratum corneum. Also, the stratum corneum is continually being shed. And if there are any microbes that are inside that skin layer, they will be shed along with those dead cells that are being shed. The skin is also slightly acidic, and that provides defense against microbes. And the skin is also salty. And as we know, that's going to provide a hyperosmotic environment that will dehydrate the pathogens that are in that area. And the skin also produces antimicrobial peptides. And these antimicrobial peptides serve to defend against bacteria, fungi, and viruses. The mucous membranes are also a first line of defense, and they produce mucus in this area. And what the mucus does is it traps the microbes, and then the mucus will be circulated out of the body, and so any trapped microbes that are in that area will be removed as that mucus is also removed. And then lastly, the resident microbes are part of the first line of defense. And as we know, resident microbes help to compete with pathogenic microbes. And they will also produce these antimicrobial substances that will make them more competitive against the other pathogenic microbes. All right, and then the second line of defense is going to be inside of the body. And a lot of these are going to be the immune cells that are there inside the body. Not all of these cells are part of the innate immunity. Particularly T cells and B cells are part of the adaptive immunity. But we're going to go ahead and go through most of these cell types here. The immune cells are divided into two main parts. We have, or two main categories. We have granular leukocytes. Leukocytes just means white blood cell. And then we have agranular leukocytes. Of the granular leukocytes, we see mast cells, eosinophils, basophils, and neutrophils. And of the agranular leukocytes, there are monocytes, T cells, B cells, and natural killer cells. Now, what we have down here are macrophages, and macrophages are just basically the same thing as monocytes. They're just more mature monocytes, and they've moved into the tissue, and excuse me, and that is where they will mature in the tissue, and that's when we start to call them macrophages. But for the purpose of this class, you can mostly consider monocytes and macrophages to be the same thing. Dendritic cells are also very similar to macrophages, and they serve a lot of the same functions. They're both phagocytic cells, but dendritic cells are localized to a certain region of the body, whereas macrophages, or more specifically monocytes, we see in the bloodstream. Dendritic cells are in a specific region. So there are a lot of dendritic cells that are localized to the skin, and they will always be in the skin unless they're activated. They're activated by coming into contact with a microbe. But again, for the purpose of this class, you can pretty much think of monocytes, macrophages, and dendritic cells as all serving very similar purposes. Then over here we have plasma cells, and plasma cells are B cells that are actively producing antibodies. So once a B cell comes into contact with a specific microbe, it will start to produce antibodies against that antigen. And once it's starting to produce these antibodies, it converts into a plasma cell. So we'll start by going through the granulocytes. And first of the granulocytes is the neutrophil. The neutrophil is phagocytic. It's also one of the most common types of immune cells. And it is one of the first ones. It's part of the early stages of infection. Then we have basophils. Basophils release histamine, and they are part of the allergic response. And eosinophils are toxic against parasites and helminths. I kind of remember this one because I think parasites are ew. So eosinophils ew, parasites ew. That's how I remember that one. All right, and then the agranulocytes. The first one is the monocytes. These guys are phagocytic. And again, remember, they mature into macrophages once they move into the tissues. And dendritic cells also phagocytic, but they are localized to a certain region. So some are localized to the skin, some are localized to the mucous membranes, some are localized to the thymus. They're localized in a particular area. But again, monocytes, macrophages, and dendritic cells, all phagocytic. And then lymphocytes include T cells and B cells, and they are part of the adaptive immunity. These three cells, neutrophils, monocytes, which are the precursors to macrophages, and dendritic cells are all phagocytic. So the mechanisms of phagocytosis. First, what's going to happen is an infected cell or a damaged cell will release chemokines. And chemokines help to tell the immune cells where to go. And so the immune cells will migrate into that region via chemotaxis. They're going towards the chemokines. And so these phagocytic cells will move via chemotaxis to the area of infection. Once there, they will attach or adhere to the phagocyte, and then they will ingest them. This process is helped by the process of opsonization. And opsonization is where a microorganism is coated with proteins. So first, the microorganism is coated with these proteins, and then the phagocyte will come along and it will recognize those proteins that are on the outside of the microbe, and that will help it to be phagocytized. And then lastly, once the microorganism is phagocytized and it's inside of the phagocyte, it will be digested inside the phagolysosome. So again, through chemokines, this is the phagocyte, the whole big part. This phagocyte will be brought into the area of infection. It will come into contact with a microbe. These are the microbes here. And then it will adhere to the phagocyte membrane. Then it will be ingested via phagocytosis, and that will form the phagosome. The phagosome will fuse with the lysosome. This is going to create the phagolysosome. Inside here, inside the lysosome, remember there are enzymes that will help to digest this microbe. So once they fuse, then in the phagolysosome, that is where we will see the digestion of the microbe. After it has been digested, then it will be discharged as waste. Now, many microbes have evolved systems to help it to evade against phagocytosis. And some of these mechanisms include M proteins. M proteins are going to be on the surface of the microbe, as well as capsules. And what these guys are doing is they're preventing the phagocyte from adhering to the microbe. And if they don't adhere, then the whole process can't happen. Other microbes will kill or lyse phagocytes through the leukocidins, leukotoxins, and they will disrupt the membrane of the phagocyte, causing it to lyse. Other microbes can escape the phagosome, so they have developed mechanisms to help them to escape the phagosome, or others will prevent the phagosome from fusing with the lysosome. And if the phagosome doesn't fuse with the lysosome, then there won't be the presence of those enzymes as well as the acidity that's inside of the lysosome, so that they won't be digested. And lastly, other microbes have developed a mechanism to allow them to survive inside the phagolysosome. And here inside the phagolysosome, they will replicate, and then they will be released when they are being released as waste. But they're not waste because they survived and replicated throughout this process. So another part of the innate immunity includes inflammation. What inflammation does is it helps to destroy pathogens, it limits the spread of infection, and it also helps to initiate tissue repair. And there are 5 main signs of inflammation. They include redness, immobility, heat, swelling, and pain. The 3 stages of inflammation. So first, there's cytokines that are released. Again, remember, cytokines are released from infected cells or from damaged cells. So these cytokines will be released. And that is what these cytokines will do is a couple of things. They will tell the blood vessels to dilate, and will also tell the blood vessels to increase their permeability, meaning they're going to allow more substances to come through. They're going to be a little bit more leaky. When the blood vessels become more leaky, that's going to allow some of this plasma, some of these fluids to leak through into the tissues. And that's where we have swelling or edema. Because the blood vessels are more permeable, that's going to allow these immune cells to pass through the blood vessels into the tissue. And so the phagocytes are trying to get into the tissue, and they will do that through margination and diapedesis. And we were going to go through both of those processes here in a second. But then through these processes, then they are in the tissue. And through chemosis, they will go to the exact location of the infection. And then pus will be formed as they are undergoing phagocytosis. And they will repair the tissue and regenerate the tissue. Older age and a weakened immune system will cause inflammation to be less effective. So here are some of the main steps. We have chemokines are going to be released. Again, remember, they are going to cause the blood vessels to become more leaky. And, sorry, what's that? Okay. So the blood vessels are more leaky, and so what they'll do is as the immune cells are going through the blood vessel, they are going to stick to the inside of that blood vessel or to the endothelium. And as they stick to this endothelium, we call that margination. Then remember that it's more permeable. And so these gaps are a little bit looser, and so these immune cells can actually squeeze through the gaps of the endothelial cells. And that squeezing through is called diapedesis. And then, of course, once they're inside the tissue where the microbes are, then they will phagocytose the microbes. Here's another picture of that. I think it's a little bit better. So again, the immune cell is being called to this area because of the chemokines that are there. And so it will adhere to the side of the endothelial cells. And what they actually do is they kind of roll along, and as they roll along, they get a little bit thinner. And then as they get a little bit thinner, then they're able to squeeze through. And this is diapedesis, as they squeeze through. So fever. Fever, of course, is a rise in body temperature. And it's actually beneficial because it will help to stop the growth of microbes. And it also helps to activate the immune system, make the immune response a little bit more effective. And this, of course, is only if it's a low-grade fever. Once it gets too high, then it can start to be dangerous for the body as well. But how fever occurs is cytokines cause the hypothalamus to release prostaglandins. And prostaglandins tell the hypothalamus to reset its temperature to a higher temperature. So antimicrobial substances, of course, remember these are substances that have effects against microbes. These include the complement system, and we're going to talk quite a bit about the complement system here in a second, so I'm just going to put that one on hold. It also includes interferons. Interferons prevent viruses from spreading to nearby cells. There's also transferins. Transferrin binds to iron. Iron is something that microbes need. They can't produce on their own. So by binding iron, it's starving these microbes. So that's what transferins do. Then we also have antimicrobial peptides, and they cause the lysis of bacterial cells. All right, so jumping into complement. A complement is a bunch of proteins that are activated in a cascade, and they have functions against microbes. So what do? They do a few things. One of the things that complement does is it will cause the pathogen to lyse. We call that cytolysis. Cytolysis, what happens is it actually is going to form holes in the cell membrane of these pathogens, and we call those membrane attack complexes. Membrane attack complexes. So these are the formation of these holes inside the pathogen cell membrane, and that causes the pathogen to undergo cytolysis. Another thing that complement will do is they can aid in opsonization. And opsonization, if you recall, increases the effectiveness of phagocytosis. And lastly, complement will help to enhance the immune response. complement can be activated in 3 ways. So this is how we're going to start complement. The first one of those 3 ways is the classical pathway to activate complement. I do need you guys to know all 3 pathways, and I need you to know all of the steps for all 3 pathways. So this is going to take some memorization and some time. So I do suggest that you get started on that early. So for the classical pathway, that begins by the antibodies will bind to the antigen. So here's the antibody in green. Antigen, of course, is on the surface of the microbe. So the antibody binds to the antigen. And that binding will activate C1. See, these are all different proteins. Okay? So once C1 has been activated, it will split into C2 and C4. Each of those will split as well. C2A will combine with C4B. Those two come together and they will activate C3. C3 will split into C3A and C3B. All 3 pathways all produce C3A and C3B. So this is the main goal of all 3 pathways, all right? So once you get to that point, they're all going to be the same. So C3A functions in inflammation, and C3B functions in cytolysis and opsonization. If you recall just the slide before, we talked about the 3 effects of complement and what they do. So these are the 2 proteins that are actually going to cause those effects. The next pathway is the alternative pathway. And in this pathway, on the microbe, are factors B, D, and P. Those 3 will bind with C3 directly, and that will cause C3 to split into C3A and C3B. Remember C3A and C3B from the slide before, and where we said that it would be the same at this point. So C3A and C3B will now have their effects. And the last pathway is started with the binding of mannose binding lectin. Mannose binding lectin will bind to mannose. Mannose is on the surface of the microbe. So this binding here is going to activate C2 and C4. And that's very similar to the classical pathway. Those 2 split, C2A, C4B, both come to activate C3. C3 then splits into C3A and C3B. And of course, C3A and C3B will have their effects. Again, here are the 3 pathways of complement activation all together side by side. What we see the exact same results, we see the same effects. What is different is what activated. Here we have the mannose binding lectin is activating complement here. Here we have factors B, D, and P activating complement. And over here we have antibody is activating complement. So again, the outcomes of complement. Cytolysis is going to create the membrane attack complex. Opsonization is going to coat the microbe, which will aid in phagocytosis. And then, of course, inflammation. In this case, with complement, the aspect of inflammation that we are seeing is the activation of mast cells, and mast cells release histamine. All of these pathways are regulated by regulatory proteins, and the regulatory proteins are going to break down the complement proteins. So again, here we have all 3 outcomes. These are the 3 outcomes. So we had 3 methods of activation, and then we also have 3 outcomes of complement. I do need you to know all 3 outcomes, but I don't need you guys to memorize all of the different steps, especially when we're talking about the formation of the membrane attack complex. I don't want you guys to spend too much time on this. I know you guys already have enough to memorize with the activation of complement. But I want you to know the main concept. So what I do want you to know for cytolysis is that it's started by C3b, and then that will activate all these other proteins to come together to form the membrane attack complex. And those membranes are going to come together inside of the cell membrane of the microbe, and it's making like a channel, an open hole inside of the cell membrane that's going to allow everything to leak out, and it will lyse. Then in opsonization, what we're having is C3b here again. C3b is going to bind to the outside of the microbe, and it's going to coat the outside of the microbe. So then when a phagocyte comes along, it will recognize this protein here on the outside of the microbe, and it will go ahead and phagocytose this microbe. And then inflammation is actually started by C3a directly, and C3b indirectly by activating C5 and C5a. So what I do want you to know is that C3a and C5a both serve to activate the mast cell, and mast cells produce histamine. Histamine is going to help to make the blood vessels even more permeable, and it's also going to serve as a chemokine. So here's just another picture of the membrane attack complex here, again forming that channel in the membrane of the microbe. And here's opsonization. So again, remember we're going to have, here's our microbe here, this yellow thing. That's our microbe, or in this case, a bacteria. And we're going to have the complement proteins that we just spoke of, or antibodies will actually also directly bind to the microbe. And so opsonization can be activated by either the complement proteins or the antibodies or both. And then because it's coated with these complement proteins and antibodies, when the phagocyte comes along, the two will bind together. So then the phagocyte can easily adhere to the microbe, and then it will phagocytose it. Some bacteria have also evolved ways to evade complement. So again, we have capsules. Capsules are going to evade complement because if we can't have the antibodies or the complement proteins binding to the outside of the microbe, then we can't have complement being activated. Some other microbes have also been able to prevent the MAC formation, membrane attack complex. So we have surface molecules that will block that membrane attack complex formation. And we also see other microbes can digest C5a. Remember, C5a was one of the proteins that's going to activate the immune response, the inflammatory response of the mast cells. And then interferons. Interferons are cytokines that prevent viruses from spreading to nearby cells. So what's going to happen here, this cell is infected. The cell is infected with a virus, and it recognizes that it's infected with virus. So what it's going to do is produce interferons. It's going to secrete the interferons, which will go to nearby cells. These cells are not infected yet. Once they come to contact with interferons, that's going to cause them to produce these antiviral proteins. So that once the virus tries to infect this cell, these antiviral proteins will degrade it so that it can't be infected. And lastly, we were talking about the transferins. Transferins, again, bind to iron. Iron is something that's needed by microbes. So if we bind to all the iron, then the microbes can't use iron, and then they're starved. And then also the antimicrobial peptides. They will inhibit cell wall synthesis and form pores in the plasma membrane. So this is also helping to inhibit some of these pathogens. And that concludes our chapter 16 on innate immunity. And I will see you guys next time.